Hey everyone, today we're gonna talk about recombining DNA, what is recombining DNA, steps of making recombinant DNA, and uses and aims of recombining DNA. They are molecules of DNA from two different species that are inserted into a host organism to produce new genetic combinations. The method is carried out by inserting a DNA fragment into a small DNA molecule and then allowing this molecule to replicate inside a simple living thing such as bacterium. The small replicating molecule is called a DNA vector or carrier, and the most commonly used vectors are plasmids. Plasmids are circular DNA molecules that replicate independently. Now let's explain the steps of making a recombining DNA. The first step in recombining DNA technology is to isolate the desired DNA in its pure form, free from the other macromolecules. Since DNA exists with the cell membrane along with other macromolecules such as RNA, polysaccharides, proteins, and lipids, it must be separated and purified, which involves some enzymes. Then an alcohol is added that causes the DNA to precipitate out as fine threads. Then it's taken out to give purified DNA. The second step is cutting DNA. Restrictions enzymes act as molecular scissors that cut DNA at specific locations. The technique agarose gel electrophoresis reveals the progress of the restriction enzyme digestion. This technique involves running out of the DNA on an agarose gel. On the application of current, the negatively charged DNA travels to the positive electrode and is separated out based on size. This allows separating and cutting out of these digested DNA fragments. The vector DNA is also preceded using the same steps. The next step is polymerase chain reaction or PCR. It is making multiple copies of DNA sequence using the enzyme DNA polymerase. It helps to amplify a single copy or a few copies of DNA into thousands to millions of copies. The next step is to cut the vector with the same restriction enzyme used to cut the donor DNA. Cut vector DNA and donor DNA are mixed in the test tube, and the complementary ends of both types of DNA unite randomly. Then now enzyme called DNA ligase, which seals or sticks the two segments, forming a continuous and stable double helix. This new hybrid DNA molecule is also called a recombining DNA molecule. In this step, the recombining DNA is introduced into a host, mostly a bacterial cell. This process is called transformation. Once inside, the recombining DNA molecule replicates like any other plasmid DNA molecule, and many copies are produced. Furthermore, when the bacterial cell divides, all of the daughter cells receive the recombining plasmid, which again replicates in each daughter cell. And now with the uses and aims of recombining DNA. First of all, recombining DNA is used to identify and sequence genes and to determine their function. It is widely used in biotechnology and medicine. Second is making human insulin. The gene for human insulin is inserted into the gap in the plasmid. This plasmid is now genetically modified. The genetically modified plasmid is introduced into a new bacteria or yeast cell. The cell then divides rapidly and starts making insulin. Third example is golden rice. Golden rice is a variety of rice produced through genetic engineering to reduce the rate of the lack of vitamin A in the world's population. Fourth example is the diagnosis of infection of HIV. HIV is the human immune deficiency virus. It's a virus that infects humans. The antibody test uses a recombining HIV protein to test for the presence of antibodies that the body has produced in response to an HIV infection. And also the BT cotton, manufacture of BT cotton to protect the plant against bollworms. And also gene therapy. It is used as an attempt to correct the gene defects which give rise to hereditary diseases. Another example is cancer. Scientists are trying to figure out through recombining DNA technology which is the gene in the genome which triggers the uncontrolled growth of cancer cells and the ways in which these genes are activated or inactivated. And the last example is mutation. Recombining DNA technology uses the method of cloning and identify that which gene is mutated. To replace the defective gene, a healthy gene of the same function is taken and multiple copies are made through cloning. And then the defective gene is replaced with that healthy gene and gene will start functioning in a normal way again.